Okay, good afternoon. Good afternoon, everyone. It's a pleasure to be here, and I would like to thank the organizers for inviting me to give this presentation. I'm going to tell you about some efforts. Good afternoon. I'm going to tell you about everyone. It's a pleasure to be here, and I would like to thank the organizers for inviting me to give this presentation. I'm going to tell you about
Cristina,
I hear an echo. What's the problem?
It's probably your tab that is open. Have a look at the top to see if there's a tab open.
Yeah, done. Okay,
okay.
Perfect.
Awesome.
Okay, so now I lost my slides. Sorry. Here we go. Okay, so I'm going to give a presentation to let you know about some efforts in my institute, NIAID, to address AFM and some early efforts to develop vaccines. Okay, so I'm going to talk about three topics. A large natural history study that my institute has initiated in collaboration with other groups. I'm going to talk about some early efforts in vaccine development, and I'm going to talk a little bit about the outcome of a workshop that we had in February on AFM preparedness. So back in July of last year, NIH awarded a large contract to study acute flaccid myelitis natural history. This was a large award that for 5 years, it was made to the University of Alabama in Birmingham to implement an international multi-study study. And there are two main PIs. One is David Kimberlin at UAB, and then is Carlos Pardo at Johns Hopkins. And the primary objective of this study is to characterize the epidemiology and natural history of the disease in the first 12 months following enrollment. And this study was initiated because there was, we still don't understand a lot about the pathophysiology of this disease and how it affects children. So one of the main, one of the secondary goals of this study was to describe in detail the clinical diagnostic evaluation and therapeutic interventions for suspected AFM cases, to identify what are the risk factors for developing AFM, to identify determinants of outcome of AFM, where some children have a more severe outcome than others, and also to describe the clinical characteristics of household contacts of patients that were being evaluated for AFM as a control. And there are, I think, 26 sites that are currently activated, and there are several sites that are pending activation, including 2 international sites, one in Peru and one in the United Kingdom. And we currently have 4 children enrolled, 2 cases and 2 controls. We didn't expect that many cases, so we might expect an increase of cases coming up in the summer. Okay, and so as we heard in the previous talks, later last year, there were several papers that pointed at enteroviruses as one of the causal roles for non-polio and for AFM. And specifically, there are 2 key papers. So we heard before from Ian Lipkin's group about the identification through some very in-depth studies about EVD68 as one of the culprits. Next slide. So as soon as our institute, as soon as we could identify at least one of the causes of AFM, our institute started to develop vaccines for AFM. And there are 2 main approaches that we are pursuing. The first approach, and both approaches are based on success that we had in the past for other vaccines for enteroviruses, mainly polio and EV71. And this to use a whole particle, either inactivated vaccine, like we have for the Salk polio vaccine. And this is an approach that we are we're planning to award a contract to develop a whole particle inactivated vaccine in July of this year to manufacture a virus and test it in a phase 1 clinical trial. And so some early work has already started to obtain new viral isolates from the CDC develops serum-free cell lines for viral production and optimize the conditions for viral growth. And the second approach is an approach that has been taken by our scientists at NIAID, the Vaccine Research Center, VRC, and they're using a VLP approach, a virus-like particle approach, where by expressing the structural proteins of the virus, they develop these the proteins self-assemble into these VLPs. And this is a schematic of what our colleagues at the VRC are doing. So they're expressing the structural protein in the red box, VP1, VP2, VP3, and VP4, in both mammalian and bacterial expression systems. And when they express these proteins, the proteins automatically rearrange into these icosahedral particles, and where the antigens arrays are highly immunogenic and they resemble very closely the normal viral particles. This is a nice approach because we have other vaccines that have been based on VLPs have been licensed. We also have the structure available of these viruses, so it's easy to compare the structure of these VLPs to the structure of the authentic pathogen. And also with some protein engineering, this technology could be applicable to related enteroviruses to make, say, a multivalent vaccine if we desire to do so. And these are some very nice cryo-UM pictures of, they compare the structure of the VLPs on the right with the structure of the virus on the left. And as you can see, they look very, very similar. They have very similar measures and so forth. Next slide. And so the same group at the VRC also is conducting some studies to better understand what is the mechanism of monoclonal antibody neutralization of EVD68. So in collaboration with another group, they identified these two monoclonal antibodies that I think Ken Tyler mentioned before in some animal studies, that these are the antibody 15C5 and 11G1. And these two antibodies, they neutralize by completely different mechanisms, where the 15C5 neutralizes the particles by basically triggering a shift in the conformation of the particle, which mimics what happens when the virus binds to its receptor and causing the release of the genome. And this antibody was found on the right top corner of the graph to be highly neutralizing in vitro. And these antibodies also were shown to be effective in preventing at least the 15C5 in increasing survival in a mouse model. So in summary, for the VLP vaccine efforts, the efforts are ongoing. They're currently testing different vaccine constructs in mice to look for immunogenicity, and also testing, they're characterizing these particles with the monoclonal antibodies that bind to the VLPs. And by the way, the neutralizing antibodies I just mentioned before, they bind very well to this VLP particle, showing that the particles, the VLPs, preserve a native conformation as compared to the virus. And the work is continuing despite the shutdown. And they're currently, it of course has been slowed down a little bit, but it's still going on. And they're currently trying to characterize the particles, optimize the structure of the immunogen, trying to figure out protein engineering to improve expression and stability. And they're also in discussions of how they could figure out more cost-effective manufacturing delivery approaches. And one of the approaches that is being discussed right now is to actually, rather than expressing the protein and purifying the VLPs, is to express the protein construct through an mRNA construct like we're doing right now for the Moderna SARS-CoV-2 vaccine. So I think we are going to see additional iteration of these vaccine efforts. And finally, I want to end with just a couple of slides on a very informative workshop that we had in February And this was a very well-attended workshop to determine what should be the research priorities on AFM. How do we catalyze the development of countermeasures, and how do we address these countermeasures? As we heard before, there are some serious challenges that I'm sure that will come up during the discussion in testing both drugs and vaccines. And these are some of the gaps and opportunities that were discussed. We need to do additional studies to really better understand the epidemiology in US and why it's changing, why we're seeing an increase in cases. We need to better understand the pathophysiology of non-polio enteroviruses mediated AFM, and more specifically, what is the role of direct viral infection versus immune-mediated damage? What is the mechanism of viral spread to CNS? And as we heard from Dr. Dugal earlier, is there a genetic predisposition to AFM that might explain why some children have a much more severe manifestation than others? You heard that there are some inactivated vaccines efforts ongoing, as well as a VLP vaccine. As you can imagine, vaccine development is going to be very challenging because it's going to be hard to conduct clinical trials to test vaccines for efficacy, since the epidemiology is so spotty and unpredictable. And you also heard a little bit about therapeutics. There are several promising antivirals that are being either repurposed or developed for, they're also broad spectrum for enterovirus drugs. It will be challenging to make sure that the drugs are delivered into CNS and the lung. And the group discussed it might be desirable to have more than one drug available. And also there are some monoclonal antibodies that are promising for potential prophylaxis and treatment of AFM, as we might hear later on from one of the speakers. And with that, I'm going to leave the stage to the next speaker, Dr. Charles Chiu from UCSF, and he's going to talk about the future diagnostic tools in AFM.